Okay, so if innate immunity is not sufficient, adaptive immunity is going to be activated. So this is that acquired immunity, and usually this takes longer, much longer to establish. And there are two types of lymphocytes, white blood cells, that are going to be playing a key role in this type of immunity. So there are T cells and B cells. So T cells are going to be involved in cell-mediated immune response, and B cells in humoral immune response. So let's take a look at T cells. So there are three different types of T cells: cytotoxic, helper, and suppressor. So we will discuss cytotoxic and helper because suppressor T cells are going to deactivate T cells and B cells when needed. So clearly when the infection is cleared, these guys are going to deactivate them because we do not want to maintain this heightened response when there is no need. So T cells are going to have a receptor that's embedded within their membrane. So take a look at this T cell here. So that is the plasma membrane of the T cell, and there are two chains of proteins. So that's alpha chain and this is beta chain. They are embedded within the membrane and they are held together by disulfide bridge. If you remember, there has to be two cysteines or cysteine on each side to be able for disulfide bridge to form. So this is that tertiary structure of a protein. So the receptor is going to have a constant region because it's going to be the same for all the T cell antigen receptors. And then there's going to be this variable region that is going to have a specific shape to be able to recognize the antigen. And when we say antigen, we do not mean that it recognizes the entire antigen, meaning that it actually binds to the epitope of an antigen. So because it's like a smaller piece, a smaller section of the antigen that binds to. But just to generalize it, it is called the antigen binding site. And so specificity here is the key. Another factor about the T cells is the fact that they do not secrete their receptors. Those receptors are permanent residents. So they can bind to antigen presenting cells, you can see in this picture, but they are not secreted, unlike the B cells. So the very first one that we want to mention is the cytotoxic T cell. So those types of cells identify infected cells, usually by viruses, because they interact with major histocompatibility complex, MHC class 1. So this protein is going to display fragments from proteins from pathogens, and those cytotoxic T cells are going to recognize them by making direct contact. So we have cytotoxic T cell, and here is the antigen presenting cell. This could be any infected cell. And there's the MHC class 1 molecule that is presenting the antigen. So you can see when there is this direct connection, now the cytotoxic T cell is going to release perforin to form pores in this infected cell, and also granzymes that are going to initiate the apoptosis of that cell, of that infected cell. So there is a dying cell. It starts to bleb. That's exactly what it does. And then phagocytic cells are going to come over and engulf the rest of the pieces and break them down and clear them out of the system. And then here's the released cytotoxic cell. It's ready to recognize another infected cell. Helper T cells are going to be triggering both the humoral and cell-mediated immune responses. So if you take a look at this particular cell, is an immune cell. This could be a macrophage or dendritic cell, and it has engulfed the pathogen, broken it down, and now it's presenting the antigen in its class 2 MHC molecule. And helper T cell is going to interact with this antigen presenting cell. And then an antigen presenting cell is also releasing cytokines as well. So the helper T cell will now release cytokines. So cytokines are going to activate, stimulate the B cells. And the B cells will begin proliferation, and eventually those plasma cells are going to be releasing antibodies into the blood, so that way those could be soluble proteins that are going to be floating around looking for pathogenic particles. Cytokines are also going to, released by the helper T cells, are also going to activate cytotoxic T cells, the ones we just talked about in the previous slide. So they can elicit direct attack on infected cells and clear them out this way. Okay, so B cells, they have to be activated, as you saw in a previous slide, they have to be activated by helper T cells. So when they are stimulated, they're going to differentiate into plasma cells. Plasma cells are the type of cells that are going to be secreting antibodies. So you can see how there's proliferation happening, and now you've got antibodies that are being secreted, and those antibodies are soluble, meaning they can travel in the blood free, looking for those particles or specific antigens to bind to and localize them. So we have different types of antibodies, and we abbreviate them in the form of Ig, immunoglobulin. So you can see right here we have different variety of those. You can pause and read and see where they are and what function they carry out. But what I do want you to know is we have a huge diversity of antibodies. And the reason that we have this diversity because of the way these proteins come together, the combinatorial joining, and there's a light and heavy chains, because those are going to be put together to form the antibody, that specific Y shape. And then when the cells are proliferating, when they are dividing, the mistakes in the DNA replication can also happen. So you may have mutations, and then the genes are going to be slightly different. That's going to give rise to a different protein. So hence, you will have that slightly different shape in the antibody. And then if you recall, we have splicing variability, which has to do with RNA splicing. So you may have this basic blueprint of the gene, you make a copy in the form of mRNA, but then the way you splice it is going to vary. So you can produce this type of protein or slightly different protein with those specific shapes that we're looking for to be able to identify specific antigens. So antigen recognition by B cells and antibodies is represented right here. So if you take a look at the antibody structure of the B cells, you're going to see that there are two heavy chains, and they are embedded in the membrane. And those two heavy chains are connected by disulfide bridge. And then there are light chains that are also connected by the sulfide bridge to the heavy chains. So, and it sort of makes this Y-shaped structure. Now, the regions that are going to vary are going to be right here, and we call them antigen binding sites. And notice we have two of them for each antibody, versus in T cells, if you recall, there's just one binding site. Now, the key difference here is that those antibodies, when triggered, they can be released. So plasma cells can release those antibodies and turn them into soluble ones. So what type of function do we have? The biggest misconception that students have is the fact that they think that antibodies kill pathogens. Well, that is not true. So keep in mind that antibodies do not kill pathogens. Instead, what they do, they mark pathogens for destruction. They capture them. They localize them. They prevent them from moving around freely. So they neutralize them. So look at this. You have a virus, and now you have a bunch of antibodies that are surrounding this virus. So yes, this virus will have a very hard time to get through the membrane and infect another cell. So most likely not going to be able to do it. So that's neutralization. So we already talked about opsonization earlier. So this is where antibodies are going to bind to antigens on bacteria, and then they're going to prepare them for recognition to phagocytosis. So macrophage is going to engulf this bacterium that's been coated with antibodies. And then opsonization, where you have complement proteins, complement system proteins that bind to the actual antibodies. So here they are. And then they promote this opsonization, as I said earlier, this engulfment of a pathogen. So that way, macrophages or phagocytic cells can better recognize where those pathogens are. So, and then as we talked about before, complement system is able to form pores in the pathogenic cells. So they embed these proteins one by one, and then when you have a pore in the membrane, obviously traffic can no longer be regulated and the homeostasis is not maintained, so the pathogen is going to be neutralized, is going to die. So how do we activate B cells in a humoral immune response? So you can see there's a lot of going on here. So we're going to begin with helper T cells, and just remember that helper T cells are activated by antigen presenting cells. So once the helper T cells are activated, they are going to be able to recognize B cells that are infected. So B cells can also be antigen presenting cells, but the way they engulf foreign particles is by, they engulf not just little tiny particles that have been already digested, they actually use, they have the antibodies on the surface, so the antibodies capture those undigested pathogens and then they go in to internalize them and then break them down. So versus if you look at a typical antigen presenting cell, such as macrophage, notice how the macrophage just simply engulfs it. So there are going to be general receptors that are going to recognize the pathogenic pattern and then engulf it. But here they use the B cells use the antibodies. So, but again, this picture doesn't show it, but the engulfment and this internalization is also taking place. So activated helper T cell directly interacts with B cell, and that causes the B cells to proliferate, meaning divide rapidly, and they divide and they produce loads of plasma cells, and those plasma cells are the ones that are going to shed or secrete the antibodies. Also, what we get from these B cells are called memory cells, memory B cells. So memory B cells are going to remain in the system, because if you get exposed to the same pathogen again, you will have a heightened response. It will be a quicker response because you're not going to have to go through all these processes again. So if you pause here and take a look in this process and see what types of cells does this process involve, you can see that all these cells are so orchestrated together. They're working together in as a group, and the ultimate goal is to clear out infection, clear the infection out of the system. So when the B cells encounter a particular antigen, what's going to happen is there's going to be this rapid division. And remember, they need to be also activated by helper T cells. So you can see we've got B cells, and they have receptors, these antibodies that have different antigen specificity. So pay attention to the shape. And then the antigens that are in the vicinity appear to be like these. So they obviously matching these patterns, and they're not matching these patterns. So it means this is where clonal selection is going to be taking place. So clonal selection means those B cells are dividing, and the reason these are dividing because these antigens are in the vicinity. So if you're going to try to make a population of B cells of this type, you would wonder, okay, what for? What are you going to fight? So that would be a waste of energy. So an encounter with an antigen selects which B cell will divide and produce a clonal population for a particular epitope. So here again, after the division, we have a group of memory cells. Those are going to stay, they're going to be long-lived, and next time you get exposed, you'll have a rapid response to the infection. And then the plasma cells, these are going to be short-lived because they're going to secrete the antibodies. The antibodies will try to capture the antigens and pathogenic particles, hopefully clear that out of the system, and these plasma cells are going to die off. So the memory, a little bit about the immunological memory. So you can see right here in this diagram, when you are exposed to a particular antigen the first time, there are going to be antibodies that are released by plasma cells. So look at their concentration levels. But then The second time when you are exposed, you will have a heightened secondary response. So you can see you're making a lot more antibodies, and you can fight the infection much, much quicker. And the reason is because you do not have to go through all of these steps. You do not have to employ macrophages, phagocytic cells, and have all these antigen-presenting cells that are going to trigger this adaptive immunity. Here, you will already have a memory cell, and the memory cell will divide into and activate and differentiate into a plasma cell, and then the plasma cell will immediately produce a ton of antibodies so that those antibodies immediately can capture the pathogens. And sometimes the response is so rapid and so quick that a person might be infected with a particular antigen, but the system clears it out so rapidly that they don't even know they have been infected. And this slide gives you a nice overview of how these cells are related. So this is just a summary of what we just discussed. So I would recommend pause here and walk through the steps, follow the arrows and see what's going on. So here it is, first exposure, what do we have? Then secondary exposure, what do we have? Differentiate between the humoral, differentiate between the cell-mediated, and look at the types of cells that are playing the role. Okay, and this is just an application. I think all of us have experienced some sort of allergic response at some point in time. If you want to follow through the steps and see how that happens, that is also going to be really helpful. So look at this. Allergen particle enters through the mucous membrane, so it enters lymphatic vessels, and then so the B cells are going to recognize these allergenic particles. And when the helper T cells make direct contact with B cells, notice the B cells are going to proliferate into plasma cells, and they're going to synthesize specific antibodies. So those antibodies are going to be embedded into the surface of mast cells. Remember, we talked about the mast cells. They have granules, and those granules are histamine type of granules that cause inflammatory response. So as the time progresses, now you have these mast cells with embedded antibodies. And if you are exposed again to that allergen, so you can see right here, the fragments are going to recognize the allergen, and then it's going to cause the mast cells to degranulate, and this is where you're going to end up having major symptoms such as red, itchy eyes, and hives, and runny nose. So that would be hypersensitivity to a certain allergen.